Hello, I hope you all had a great lunch. My name is Song Wei-Im, one of the co-presidents of Wisdom, and I'm delighted to announce our next speaker, Namrata Anand. Dr. Anand is a bioengineer, computer scientist, and founder of Diffuse Bio, working on problems at the intersection of machine learning and biology. For the past several years, she has been working on re-envisioning the computational protein design toolkit with AI. Some highlights include the first crystal structures of AI-designed proteins and diffusion models for protein structure generation. Today, she will be discussing her research in building generative AI systems for protein engineering and protein design. She will also reflect upon her Harker experience and her trajectory since graduating in 2010. Please welcome Dr. Anand.
First of all, thank you so much to Ms. Chetty for organizing this event and for inviting me. And also wanted to say thank you to all the students and parents and teachers who put this event together. I'm an alum. I graduated in 2010. And yeah, it's a huge honor to be back here. And it's kind of crazy to be back after so long and seeing kind of all the changes to the campus. So it's really, really exciting. Also just want to quickly say thank you to Alex Ren and Vijay and the Fellows Fund team for suggesting that I speak, and also for supporting our startup, Diffuse Bio. And yeah, I'm super excited to talk about some of my research as well as what we're working on at Diffuse. Title of my talk is Generative AI for Protein Design, and you know, it seems like there's the topic of the symposium is the AI revolution. And AI is really changing so much. There are a lot of natural language breakthroughs with models like ChatGPT, image generation. But I'll be talking about something a little bit different, which is AI for molecular design and for basically solving problems in science. So yeah, just a brief outline of what I'll be talking about. First of all, I'll just go over a brief background about the problem. We'll go over some results over the past several years and talk a little bit about the most recent breakthroughs around a particular class of models called diffusion models. Talk a little bit about our startup, Diffuse Bio. And finally, I'll spend a little bit of time talking about my path going through Harker, starting as a small third grader all the way to graduation, and you know, roughly 15 years since. Cool, so for starters, you know, what are proteins? Why do we want to design them, and why should we use AI to design them? So, you know, I think when I talk about proteins, for biologists, we have a very clear sense of what, you know, proteins are, but I think for most people, you might envision, you know, the food that you eat, you know, beans, legumes, and other things like that. Or you might think about, you know, the protein shakes and protein powder that you have after a workout. And that is pretty much what a protein is. Proteins are molecules that have an incredibly wide variety of functions in our bodies. When we eat a source of protein, it basically is broken down into small building blocks, which are then reassembled into molecules that do a whole range of things in our bodies. You know, they catalyze chemical reactions. You know, they help our muscles function. And beyond that, there's, you know, so many things that proteins do. And these are just molecules that do some amazing, amazing things. And if you zoom into, let's say, the nanometer scale, what we're talking about is a molecule that looks something like this. It's basically a repeating series of amino acids, which are these building blocks. And proteins are basically this backbone with side chains branching off. So the protein can be thought of as a chain that will then fold up into a three-dimensional structure. that 3D structure has some ordered components that look like these helices, as well as these beta strands, flat kind of sheet-like regions. And so we can think of proteins as basically this type of a chain that folds into a 3D structure, the building blocks of which are amino acids. We can also think about proteins in a couple of different ways. We can also think about proteins not just as a molecule that occupies some 3D space, but a chain that actually torques and has some amount of torsion and folds in on itself. We can also think about the protein as a string of characters. This might just look like a random string, if you're typing on your keyboard and you type some random characters, but what we're seeing here are basically the amino acids. So every single character here corresponds to one of 20 canonical amino acids. So proteins can be thought of in these different ways conceptually, and they have a ton of different functions. But why do we want to design them? The potential here is to be able to design new therapeutics, new enzymes, new diagnostics, new vaccines. If you guys remember back, during the pandemic, people were talking about monoclonal antibodies as a therapeutic, as basically a first pass way to kind of block the viral infection. That's a protein that we're talking about. Enzymes, you could imagine being able to design molecules that can break down plastids. Diagnostics, you could imagine having really rapid diagnosis of diseases. And a lot of vaccines are basically protein subunit vaccines. They're basically just a protein that's injected into our arm. And so, as I was basically thinking about my own journey and kind of what research area I wanted to get into, it seemed to me that design of these molecules was the bottleneck for so many incredible potential problems that I wanted to work on and help solve. And so, design of proteins on a computer, this is not a new problem. This is a decades-old problem that people have been working on. And there have been some remarkable results in computational protein design, things like new enzymes. So over here, what I'm showing you is some work by scientists that created this molecule that when folded, would do this chemical reaction, catalyze a chemical reaction called Kemp elimination. It's a proton transfer reaction. And the really cool thing about this is that there's actually no naturally occurring protein that can do this reaction. This is a protein that's been engineered that can do a chemical reaction that's not at all seen in nature. Other examples of really cool results over the years, this is scientists were basically developing a therapeutic against influenza. So this is a protein that will actually find a receptor binding site on the hemagglutinin protein and neutralize the virus. And here's an example of a biosensor development project where they designed this protein that could basically capture and bind a steroid molecule. So there have been some really remarkable results. However, a lot of these results are limited to modifying naturally occurring proteins. So you've got to take some existing molecule and try to modify it and change it in order to re-engineer it for a different purpose. While there have been these great results, unfortunately these results are, I would say, few and far between. Each of these projects has taken teams of scientists to basically work for several months or years at a time in order to get these results. And the reason behind this is because the kind of standard computational protein design toolkit has successes, but there are ways in which it's unreliable. So how do people currently design these molecules? Well, you can imagine that if I want to design, let's say, a new protein, what I could do is think about the individual atoms that are in this molecule and model the energetics between those atoms. So model how, let's say, between a nitrogen and an oxygen atom, there might be van der Waals forces, there might be electrostatic effects, there might be hydrogen bonding. So what I can do is write down basically a physics-based model where I model essentially the energy as a function of the distance or relative orientation of these atoms. So for instance, I can talk about some kind of repulsive force between two atoms or some kind of attractive force. I can talk about the hydrogen bonding between basically sets of atoms. And while there's been decades of work that have gone into modeling these kinds of energy functions and they're pretty mature, they have a couple of trade-offs. One is that they're slow. They can take hours or days to optimize. And then the other is what I was alluding to before, which is they don't work all the time. And it's pretty hard to actually get a successful molecule out of doing this type of design. And so around 2016, when I was getting into my PhD, I was really interested in deep learning and machine learning, but I was also really interested in science. And I kind of saw protein and molecular design as being really a problem that if I managed to kind of solve or make headway on, I could do so many more cool things in science. The crazy vision that I had was being able to design these really large molecular machines. If people have heard about CRISPR and gene editing, that's basically a protein that binds a guide RNA and basically can cleave or edit DNA at a selective location. So I had this vision of just why do I need to try to discover a molecule that does gene editing? I should be able to just engineer that type of molecule from first principles. And so I was kind of in the right place at the right time. I was really interested in deep learning. I had kind of spent a lot of time building computer vision models and natural language models, but I had also spent a lot of time in the sciences and trying to kind of think about scientific problems. And so it was very natural for me to kind of put these two things together and say, hey, why don't we use deep learning to design proteins? So I know the topic today is AI revolution, and so I'm sure deep learning has come up more than a few times already, but just a quick primer on what exactly deep learning is. So deep learning, in maybe the simplest instantiation, is a deep neural network, which can take in some kind of input and produce an output. And that network basically encodes a complex nonlinear function. So in this case, what I want to do is I want to take, a picture of, let's say, George Washington and predict that, the guy's name is George. And so what I can do, given enough data where I've got input and output labels, I can train this neural net to actually reliably predict, given the picture of, one of the presidents, predict his name. And the way to think about this is you basically have, the network is made up of parameters or numbers, and you have some kind of objective function. In this case, I want to map between an image and a label, and you can basically try to optimize the parameters of the net in order to better predict your label. And so this idea, this idea of training these types of networks turned out to be incredibly powerful. Up until we were doing this type of modeling, what people would do is, let's say I wanted to do this image classification task. What people would do is they would take this image, think about really clever ways to extract information from that image. So for instance, say, okay, maybe I could come up with some kind of edge detection algorithm or some way to pull out some features from this image that could help me classify, which president it is. And it turns out that deep learning has pretty much surpassed all of those really clever modeling approaches. And so my thinking was, we're doing a pretty similar thing here with molecular design. We're thinking really deeply about the physics, we're writing down these complex mathematical models, but in other areas, deep learning pretty much obviates the need for some of this more complex mathematical modeling. And I'm sure you guys are super familiar with a lot of the ways that, a lot of the areas that deep learning has already revolutionized, whether it's autonomous vehicles with Tesla, autopilot, and other autonomous systems where now we can basically just have a camera and there's a neural net just like this behind the scenes that's reasoning about what's in the scene. There's AlphaGo, which is a reinforcement learning algorithm that does better than human performance on the game Go. How many people here have heard of AlphaFold? Okay, so for those of you who haven't heard about AlphaFold, it's basically a team from DeepMind trained a model to go from protein sequence, so that sequence representation I was showing you, to predict the 3D structure. And this was a huge breakthrough in our field, basically surpassed by an order of magnitude everything that had come before. And I'm sure everyone here has played with ChatGPT and DALL-E. And so, deep learning is the engine behind all of these models. And while, intuitively it should be applied to things like natural language or computer vision, for me, back in 2016, it was a natural thing to apply it to molecular design. And so this was kind of the crux of my idea, almost 8+ years ago, which is to use generative AI to design proteins. And I'm sure everyone here is super familiar with DALL-E and ChatGPT, but the equivalent. So let's say with the case with DALL-E, what I'd like to do is give the model a prompt, so something like a teapot in the shape of an avocado, and the model will decode many possible realistic images that correspond to that prompt. And similarly with ChatGPT, I would like to give it a prompt. So in this case, explain to me how AI is going to change how we design proteins, and the model produces very realistic text as an output. So the analogy to what we're trying to do with molecular design is, I'd like to, as a scientist, be able to tell the model, I want a protein that has this particular shape, or I want a protein that catalyzes this particular chemical reaction, and the model should decode many, many possible solutions to that problem. So that's just the analogy between what we're doing compared to ChatGPT or DALL-E. And just very quickly, how many people here have heard the term generative AI? Pretty much everyone, right? So one thing that distinguishes generative AI from, let's say, kind of predictive AI models, right? I thought actually maybe useful to just briefly go over this. So there are basically AI models that will do things like, if you give it an image of a cat, it will produce a label that says cat. Those models are called discriminative models. So basically, if I've got some input data, let's say in this case it's an image, and I've got some output label y, in this case a label that says cat. A discriminative model is modeling the probability of the label in the input. So the model ideally will tell me, if I send in an image X, it'll tell me the likelihood of label cat versus label dog versus any other label. In contrast, a generative model is trying to model the actual data distribution itself. So the generative model is saying, I would like to just produce images that look like input data X. So if I were to train a model on input data that looks like this image of this cat, then ideally every time I draw a sample from the model, I can get some kind of realistic image. The types of models that we're talking about here with DALL-E and ChatGPT are conditional generative models. So what this means is, rather than just sampling, some random image X, I would like to sample an image conditioned on a label or conditioned on some input prompt. So let's say in the case of DALL-E, conditioned on the label a teapot in the shape of an avocado, I now want to decode many samples, many images. And so what we're doing is, with design, right? AlphaFold, for those of you who are familiar with it, right, is a discriminative model. It's basically saying, given the sequence of the protein, predict the structure. But a lot of our kind of bioengineering tasks of interest are actually design tasks. They're tasks where we want to actually sample many putative outputs that can actually solve a given problem. Let's say with therapeutics development, I want to sample many, many, many candidate therapeutics. I don't necessarily just want to predict a single output. And so this is the approach that we've been taking to design new proteins. A couple of other things to just point out is, these models seem to have the capability to almost extrapolate beyond the data it's seeing. And we don't really know what's happening here, whether it's some kind of simple interpolation between the data, the input data, or whether there's some kind of non-trivial generalization or extrapolation happening. But it does seem like these models are capable of seeing some input data and then producing something that's substantially different from the input data that it's seeing. The other nice thing about these models is you get an output in seconds rather than running some kind of complex optimization procedure for hours or maybe even days in the protein design case. So all of this for me was the motivation behind tackling this as a research problem that I took up in my PhD. And so what I'll do now is I'll just walk you guys through an evolution over what kind of 8 years of research looks like for this particular task. So starting off, the very first attempt at generating new proteins, I started in 2016, and it was a very simplistic approach. It's the idea that if I want to produce something that's got coordinates in space, I can represent those coordinates as the distances between every pair of coordinates, basically a pairwise distance matrix. And so the idea here was to take what's known as a generative adversarial network and use this type of network to produce these pairwise distance matrices which correspond to structures. So very quickly, because I think it's interesting maybe for the younger students, the audience who are interested in building these types of models, this type of GAN network is actually pretty easy to implement these days with the frameworks we have. So it's actually a nice starting project for students who are interested in kind of trying things out with PyTorch or TensorFlow or the existing frameworks. The idea here is that you take a generator network, you ask it to produce a fake example. So in this case, it's producing a fake pairwise distance matrix corresponding to some protein structure. And then you take a discriminator model, and the discriminator model's task is to differentiate between real and fake data points. And so when you train in this way, the generator and discriminator are basically almost in competition with each other. The generator is trying to fool the discriminator, and the discriminator is trying to basically guess which samples are actually coming from the generator and which are coming from your actual input data source. So what you can see here on your right is basically some real fragments from protein structures. These are very short fragments that are basically excised from existing 3D protein structures. And what we can get from this type of early generative adversarial network. And what you can kind of tell is maybe qualitatively they match. There's some helices being formed, there's some beta strands being formed. But overall, these are pretty short fragments. They do have errors, they have clashes. They're not very highly realistic. And so really the challenge, not just for protein design, but really with generative models with the entire field for the past several years, has been to try to get the outputs of these models to be more and more realistic. And so that was what I was kind of doing in all my subsequent work, was just trying to get the models to perform better. A couple of other things, so we were able to kind of show that if you have this type of generative model, you can now start to optimize these backbones and move them around in a really efficient and easy way. But as you can kind of tell, right, from this simulation, this is showing you every atom on the protein backbone. There are some clashes. It's not very large, right? It's unclear. It's not really forming a nice globular structure. And so there was a long ways to go. A couple of other things that we were working on is another task that you can think about is, if I've got this structure, can I find that sequence of amino acids that will fold that structure? It's basically what's called the inverse protein folding problem. It's the inverse of what defines it with AlphaFold. It's the idea of, can I find, if I've got this type of structure that I want, to fold, can I find a range of sequences that will fold back to that structure. And so what was really cool for me, kind of from my personal experience, is we trained these models, we then tested them in the lab, and we're trying to basically show that we could get an AI-designed protein to fold as designed and validate that. So what you're seeing here is basically a schematic of the crystal structure of a barrel-shaped protein that we designed in this way, where it's overlaid with the design model. So we were able to show that we were able to very reliably get these design sequences to fold back to the structures that they were meant to fold into. So that was a cool result. But I still felt like this problem of being able to design things that just from scratch that look like whatever you can dream up, that we still hadn't hit that goal. And so cut to early 2022, I was playing around with this class of models called diffusion models. Who here has heard of diffusion models? Okay, cool, awesome. So diffusion models are they're basically the model behind all the image generation models that we're seeing right now, DALL-E and Stable Diffusion. I'll go into a little bit about how diffusion models work, but very excitingly, once we basically adapted those models to this domain, we were able to get large coherent protein structures. And what you're seeing here is basically samples from a generative model. And this is the tiniest possible model you can imagine. This is something I was training at home on a single GPU, super, super inexpensive. But just by kind of like the algorithmic breakthrough, we were able to get results that were just far beyond what anyone else in the field had shown. So this was a super exciting and fulfilling moment for me because it had been years of kind of working pretty single-mindedly on a research problem that I thought was super important, and then kind of getting through the breakthrough moment. So yeah, it was just a super cool moment for me as a researcher. Just a brief intro into diffusion models. I think one thing I just would want to say to the students here who are interested in AI is, you should be pretty undaunted to just kind of try to build some of these models yourself. I think you'd be surprised at kind of how easy it is to actually get maybe a first-pass model working. Really what you need is, you need to know a little bit of coding, you need to know some Python, you need to know some calculus and some linear algebra. And it's also a good idea to read a lot of the papers that are coming out and thinking about, how can I maybe try to implement this? But a lot of the models that are some of the top performing models today are actually quite simple. Of course, people get amazing performance by scaling them up enormously, and pretty sure no Harker student has a huge GPU cluster sitting at home. If so, let me know. I think it would just be cool for students to kind of feel like they can maybe just jump in and try to implement some of these things. So yeah, coming to diffusion models, the simple premise here is that the idea is that I want to take this model that I'm training and give it, let's say instead of giving it a perfect image, in this case an image of a face, what I'm going to do is add some noise to that image. So I'm going to take that image, I'm going to add some noise to it, I'm going to take some of the pixel values and corrupt them slightly. And then what I'm going to ask the network to do is take that corrupted image and try to predict essentially the original image. With diffusion models, they do this a couple of different ways. You can either try to predict the noise that was added to the image, or you can try to predict the actual underlying image. But very simply, that's what the network is tasked with doing. And what you can do is you can add different levels of noise to the image. So you could imagine that if I added just a tiny bit of noise, it's quite easy to determine what the underlying face should be. But if I add a ton of noise, I might actually destroy all of the information that's in the image, making it almost impossible to actually predict anything about the underlying image. The idea is to train the model in this way, such that when you are trying to draw a new sample, meaning I enter a prompt into DALL-E or Stable Diffusion, and I want to decode an image, what it starts with is just random noise. And what it's trying to do is condition on the prompt that you give it, try to kind of figure out what underlying features are in the image and predict some kind of underlying ground truth. And you can do this kind of decoding in several steps. So the model has, it doesn't just have one shot to produce an image, but it has several iterations to try to improve the underlying image. And so this is a paper from John Ho in 2020, which was, I think, the first major result showing diffusion models working on images. None of these people actually exist. These are all generated images from training on a data set of images of faces. And so, when I saw this result, I thought it was very natural to try to adapt these models to protein design, because whether or not you're trying to generate an image, or you're trying to generate a text string, or you're trying to generate a molecule, the core of it kind of remains the same. It's these deep learning models that have enabled us to build really strong generative models. So basically, there were a couple of kind of algorithmic insights needed to adapt these diffusion models from operating on a 2D grid of pixels, which is an image, to now operating on this 3D, these 3D points in Cartesian space. A couple of things, I won't go into too much detail, but basically, if you have a molecule that's in 3D space, if I rotate it or if I move it, it's the same molecule. So I need to make sure that whatever model I'm using sees and understands that a rotated molecule or a translated molecule is in fact the same molecule. And so that was the kind of trick necessary to get these models to work. But you guys can, compare these outputs where these are large structures, they're globally coherent, they basically don't have any errors. If you compare that to going back to my first attempt at this in 2016, these look substantially different. And it's just been cool to see because, in the image generation field, we've seen a similar trajectory where, 8+ years ago, the generative models were producing, very noisy, very, very improbable images, and now we're seeing amazing performance from these models. So it's been kind of cool to see my own kind of version of that in this domain. I showed you guys this barrel structure, which came from essentially starting with a protein structure that looks like this barrel, and trying to find a sequence that's going to fold back to the structure. But now what we're able to do is actually generate that starting structure ourselves and the sequence as well. So what I basically told the model, in this case on the right, is I would like you to produce a structure that has a barrel in the center and 8 helices surrounding it, and I also want it to be 4-fold symmetric, and I also want a sequence that's going to fold into it. And we can basically decode molecular structure that is at that level of detail and accuracy. So that's basically where the tech stands today, and what that's brought me to is basically starting a company, which is Diffuse Bio. If you guys are interested in learning more about us, our website is diffuse.bio. But once we saw these empirical results, it was clear that the tech had actually matured to the point where it could actually be the basis for starting a company. And for us, our motivation is to further develop these models to design essentially functional proteins that people need. This can range from therapeutic molecules, enzymes, biosensors, diagnostics. I think the potential is absolutely enormous, and the cool thing is, it's been a long process to get to this point, but the entire field has advanced, I think we're working on some cool things. And so just to talk a little bit about the company, what we're doing is we're scaling up our generative models, and we're also developing biological assays to actually try to generate orders of magnitude more data. So deep learning models, as you guys may know, are super data hungry. And if you think about OpenAI and GPT-4, these models are being trained on, as much of the internet as these engineers can scrape. In our space, we have a lot of data, but we don't have internet scale level data. So another big thing for us is to actually try to generate data at the right scales to be able to train even more powerful models. So that's a little bit about diffuse. And let's see. Okay, cool. So yeah, if you guys want to reach out, our email is info@diffuse.io, or you can just go to our website. Would love to hear from folks who are interested or have any feedback for me, or even with students, we have a couple of positions for interns if people are interested. So definitely reach out. Cool. So just in summary, I think there's incredible potential for computational protein design. It is a little bit different than natural language or robotics, these other areas, but I think the potential impact is going to be really, really big. Standard methods have been slow and pretty inaccurate, and we're already at the point where generative AI systems are producing better quality designs and significantly faster. And it's still early days for this type of tech. I think one thing that a lot of young people that I talk to who are getting into ML tell me is that with LLMs, it kind of feels like maybe the problem almost feels solved. I mean, I still think there's a lot of interesting problems within natural language, but it feels like some of these large incumbent companies that have billions of dollars have basically solved the problem and are just going to win by scaling up these models. And so a lot of young students who are in undergrad or in their masters have basically asked me, what should we be doing if we're interested in AI? And I think it's cool to work in a domain that's a little bit more nascent and where there's still a lot of work to be done. And so I think whether it's this domain, there are other domains, whether it's material science or other kind of vertical areas where you're kind of putting together AI plus some domain knowledge that you've developed. I think that's going to be a super, super exciting and kind of fruitful area. So yeah, that's kind of it for the technical part of my talk. But I thought it might be interesting, especially for the students, the Harker students, to hear from now kind of a more ancient alum about kind of what it was like to be a Harker student way back in the early 2000s. Kind of what my trajectory has been since then. So, in the morning before this talk, I was looking through some old photos because I thought it might be cool to just kind of look back at the whole kind of history. And I found these cool pictures. I found basically when I was joining Harker as a third grader. And one thing I'll say is that, I think Harker, we have an incredible community here. And all the students here will know there are countless opportunities, to explore all of your passions and interests. And, for me, when I was in second grade, my mom was applying to be a teacher at Harker. And my mom actually has taught at Harker for 20 plus years as a first grade science teacher. And she's right there. And, I remember as a kid, I'll show you guys this other picture. This is Grandparents' Day, 2000. And this is me with my cousin Gautam and my sister Anand and our grandparents and great-grandparents. And it was, as a second grader, my cousin Gautam was already at Harker. And so we had heard about Harker. And I remember my mom telling me that, Harker has field trips. And you can go to Montreal Bay Aquarium, you can go to Coloma, you can go to Marin Headlands, you're going to be able to go to Yosemite. And that was just mind-boggling to, a second grader. I thought that was the coolest thing in the world. And we were so nervous and just really, hoping that we'd get into Harker. And getting into Harker was honestly like a dream come true for myself and my sister. And so I'm just super grateful to the school. And I think for those of you who are here, you have so many opportunities at your fingertips. And I would just maximize the opportunities and just enjoy the ride. Harker being a place at the heart of Silicon Valley, there are a lot of very successful parents and lots of expectations on students, and that was the same for me as well. But I think it's sometimes it's good to put those expectations a little bit aside and just focus on discovering who you are, what your passions are, and also to rest kind of easy knowing that you've got an amazing network to tap into, an amazing community. And in the process of starting this company, I think there have been so many Harker alum that I've met who have applied, investors who are affiliated with the school, parents of the school, and the community and the network has been invaluable. So to all of the kids out there, I would just say, keep hold of those friendships and that connection, and just maximize the opportunity while you're here. I was, very lucky to be an Intel Science Talent Search finalist, and I still remember when this happened back in 2010. I was completely shocked. I had done summer research for about 2 months the summer before my senior year, and I got to work with this amazing scientist, Dr. Rajat in astrophysics, and I really didn't know anything. And I played around with some data and learned as much as I could. And what I realized, it was kind of mind-boggling to win this award. Of course, I was really happy and excited to, but I think this experience, and this is myself and Dr. Schaefer, who was kind of working on the research program with the students at the time. And what I realized, the experience of kind of winning this almost by kind of happenstance and chance is that, awards and accolades, I think, are really, great to strive for, especially if it motivates you. But ultimately, and pretty much if someone wins an award, 9 times out of 10 is very well deserved, and people have put in the hard work and they deserve to be kind of achieve that. But awards are also a little bit arbitrary, and there's a lot of luck involved in many things. And I think my unsolicited advice to the young kids would be to focus on your passions and focus on your own kind of growth and learning, and don't worry so much about the accolades and the acknowledgement, because if you feel satisfaction in what you're doing, that at least in my experience makes me feel great. Just continuing on the unsolicited advice portion of this talk. I think another thing is we probably have some seniors or juniors in the audience who are going to go off to college. And one thing that I think has served me particularly well, but also a lot of my classmates who have gone on to do really incredible things, is really using this time in high school and or elementary school, middle school, wherever you are, and beyond in college, to explore as much as possible. Right now with AI, it can be very tempting to just want to become an expert in AI. But I think you'll benefit tremendously from kind of exploring things even beyond the scope of AI, and maybe even think about how to kind of integrate AI in different areas. So just to give you guys a sense, I switched my major 3 times. I thought I was going to go to physics, then I switched to chemistry and biology, and then I switched to econ for a little bit, and then I ultimately switched to applied math. Maybe that's 4 times. You guys should account for me. I switched majors a lot, took a lot of different classes. I went and tried different things in different labs, 3 different labs. I took a summer off to pursue music and study music in more depth. I also worked on a startup toward the tail end of undergrad. And most importantly, I had a lot of fun and did a lot of things that were not super productive. So I would say that, in that process, I learned a lot about myself and how to think, and what types of problems I'm suited to working on, and what I enjoy. I realized that I really enjoy coding. I really enjoy tinkering and building things and experimenting with things. And I found a path for myself to put those things together. So my unsolicited advice to the kids out there is to, explore and really figure out, who you are and what your passions are. So I think maybe in the interest of time, maybe I'll just spend a minute on this. I think it might also be helpful for me to talk about kind of how I picked a research problem and how did I come across this area, because It's been tremendously helpful when I've been trying to fundraise for the company or trying to recruit, that I was the first person to do this and had an established kind of research track record. But how did I kind of figure out how to even do this? And it was very much an exploratory process. I was in this lab working with this really amazing scientist named Adam Cohen, and he was doing a lot of work in optics and neuroscience. And so they were interested in studying the junctions between neurons called synapses via optical imaging. And so basically they wanted to leverage the fact that some proteins can basically fluoresce, they glow. If you guys have heard of GFP, the protein actually fluoresces. And what I was tasked with doing as an undergrad is finding a fluorescent protein that had a particular pH dependence of fluorescence, or rather photoconverting between two fluorescent states. And as an undergrad, I was like, how the heck am I supposed to find this protein that has some kind of magical pH dependence on fluorescence? And I scoured the literature and was trying to understand, how do I find this? And that's when I kind of, after a couple of weeks, a couple of months of not being successful, I kind of thought, what if I could just try to build some kind of machine learning model to predict fluorescence from the protein sequence? And at the time, with the models that we had, SVMs and very classical basic ML, this was kind of even before neural nets were kind of in the lexicon and widely used, people thought I was nuts, because I kind of was, because with these small models, with these very simple models, it was not a well-posed problem. But that was the seed for me, that I want to build a model that can do this type of prediction. And that's ended up being I guess since 2011, it's been a more than a decade plus long journey of being really passionate about ML for protein design and for protein engineering. And so that was kind of the seed for me about being interested in ML and AI for solving scientific problems that you maybe can't really reason about from first principles. So I thought that might be helpful as well, that if you try a bunch of different opportunities, you might kind of see some connections happening that end up putting you on a great track, even if you're a little bit ahead of everyone else. So yeah, that's kind of it. For anyone who's interested in even more unsolicited advice, definitely come find me. I have a lot of things I'd love to share with students who are kind of on the cusp of high school, college, or even earlier. But with that, happy to take any questions, and thanks so much for your time.
Thank you again for your talk about computational proteins and your journey at Harker. Now we have the opportunity for a short Q&A. So if you log on to the ConnectMe app, you can enter questions. Our first question is, what is one of the hardest challenges that you've gone through, and how did Harker help you discover any of your interests?
Hardest challenge, I think one thing that Harker does a great job with is preparing us for handling any kind of academic challenge. But it turns out that in the real world, a lot of times challenges are not the technical challenge that you're trying to solve, or the code that you're trying to compile. Oftentimes challenges, I feel, are interpersonal in nature. And so my toughest challenges have been with dealing with people that don't see eye to eye with me, or that just have a different perspective. And yeah, I think, what was the tail end of that question? It was, how did Harker help? Yeah, so toughest challenges. Yeah, I would say that for me, I think one thing that Harker helped me with is I did a lot of performing arts when I was here. And so I think that was actually a good foundation for learning how to relate to people and kind of trying to understand the human condition. So that's been great for me. And and Harker, I think just naturally, within the amount of opportunities, the abundance of opportunities, it's very natural to find your interests. I was talking to some of the WISTEM girls over lunch, and they do dance, and they do debate, and they do journalism, and it's, I think the nice thing with Harker is you can kind of explore and find what you want to do.
We have one more question. How do you come up, what are some things you would recommend younger students do to get introduced to the many large scientific topics that you covered and the kind of computational protein modeling field in general?
It can be daunting to kind of try to understand all of the major scientific problems that people are tackling today. I think a good place to start is to just learn about one, whether it's finding like a research opportunity and going to a lab and listening to people talk. The first few weeks for me, whenever I would join a new lab, it's like they were speaking Greek. I did not understand, for the first few weeks. It takes some time to understand, but yeah, I would start small, right? I would start with a couple of problems that pique your interest, and then just reading widely and talking to people. I think is the best way to learn this type of thing. Cool. Thank you all so much.
Thank you so much, Dr. Anand, for the amazing keynote address. It was really inspiring as a current student, and I'm sure for many other members of the audience, to be able to hear and learn from such an accomplished alumni. So let's give one more round of applause to Dr. Anand.